Hello everyone, welcome to Groups Master. This is APPSC Group 1 Mains Revision Series Science and Technology. వీడియోలో మనం టాపిక్స్ 8.3 and 8.4 డిస్కస్ చేస్తున్నాం. ఇంట్రడక్షన్ టు జెనెటిక్ ఇంజనీరింగ్ అండ్ బయోటెక్నాలజీ అండ్ బేసిక్ కాన్సెప్ట్స్ ఆఫ్ జెనెటిక్ ఇంజనీరింగ్ మనం ఈ వీడియోలో చూస్తున్నాం. దిస్ ఈజ్ అవర్ అఫీషియల్ వెబ్సైట్. ప్లీజ్ విజిట్ దిస్ వెబ్సైట్ ఫర్ ఫర్దర్ అప్డేట్స్ అండ్ ఇన్ఫర్మేషన్. Let us see biotechnology. What is biotechnology అనే క్వశ్చన్ వస్తే ఈ విధంగా ఆన్సర్ చేయొచ్చు. Biotechnology refers to technological process that modifies any living organisms or processes to develop or create different products that serve specific purpose. Simply, biotechnology refers to genetic manipulation of organisms for specific purposes. Biotechnology has two main branches. One is tissue culture, another one is genetic engineering. Tissue culture అంటే ఏంటంటే it refers to the in vitro cultivation of cells, tissues, organs, embryos, seeds in a laboratory. Genetic engineering ante it is the process of manipulating the genetic makeup of an organism in order to change its desired characteristics. Biotechnology applications already in the previous video lo chusam, aa video ni refer cheyochu. Next, genetic engineering. Genetic engineering gurinchi ఏ క్వశ్చన్ వచ్చినా ఈ విధంగా ఈ పాయింట్ యూస్ చేసుకొని ఇంట్రడ్యూస్ చేయొచ్చు. It is the process of manipulating the genetic makeup of an organism in order to change its characteristics. This is typically done by altering the DNA of the organism either by adding or removing or replacing the specific genes. Recombinant DNA technology is used to modify an organism's DNA to achieve desirable traits. This is revision series, so చాలా స్పీడ్గా మూవ్ అవుతాం. It involves adding a gene from one species to an organism or from a different species to produce a desired trait. An organism that receives the recombinant DNA is called genetically modified organism. Shortcut lo GMO antam. If the foreign DNA that is introduced comes from a different species, the host organism is known as transgenic. In genetic engineering, DNA of living organisms are manipulated to produce desirable traits or to remove unwanted traits. It is typically done through a variety of techniques, including gene editing, gene insertion, gene deletion. The process of genetic engineering begins with the isolation of specific gene or set of genes that are responsible for a particular trait. These genes then be modified using various methods such as cutting and pasting DNA sequences or introducing new DNA sequences from other organisms. ఈ విధంగా జెనెటిక్ ఇంజనీరింగ్ గురించి క్వశ్చన్ వచ్చినప్పుడు ఈ పాయింట్స్ అన్ని యూస్ చేసుకోవచ్చు. నెక్స్ట్, జెనెటిక్ ఇంజనీరింగ్ హ్యాస్ మెనీ అప్లికేషన్స్ ఇంక్లూడింగ్ డెవలప్మెంట్ ఆఫ్ న్యూ క్రాప్స్ with improved yields and resistance to pests and diseases, the production of pharmaceuticals and other useful components, and the creation of genetically modified animals for medical research. Ivi applications. Genetic engineering has been applied to the production of cancer therapies, genetically modified plants and livestock, and many more. While genetic engineering has potential to offer many benefits, it also raises ethical and safety concerns, particularly when it comes to the release of GMOs, genetically modified organisms, into the environment. As such, there are strict regulations governing the use of genetic engineering techniques in many countries around the world. ఈ విధంగా జెనెటిక్ ఇంజనీరింగ్ గురించి ఏదైనా క్వశ్చన్ వచ్చినప్పుడు పాయింట్స్ యూస్ చేసుకోవచ్చు. ఆల్రెడీ జెనెటిక్ ఇంజనీరింగ్ అప్లికేషన్స్ ప్రీవియస్ వీడియోస్లో ఉన్నాయి, ఒకసారి అది వెళ్ళి రిఫర్ చేయొచ్చు. నెక్స్ట్, బేసిక్ కాన్సెప్ట్స్ ఆఫ్ జెనెటిక్ ఇంజనీరింగ్. కొన్ని బేసిక్ కాన్సెప్ట్స్ చూద్దాం. ఫస్ట్ ఆఫ్ ఆల్, వాట్ ఈస్ DNA? DNA is the genetic material that contains the instructions for the development and functioning of all living organisms. It is composed of nucleotides, which are the building blocks of the DNA. Next is gene. A gene is a segment of DNA that codes for a specific protein or RNA molecule. Each gene contains instructions for particular trait or characteristic. Genetic code. The genetic code is the set of instructions that determines how nucleotides are arranged in a DNA sequence, which in turn determines the traits of an organism. What is recombinant DNA ante? Recombinant DNA is a DNA that has been artificially modified in a laboratory. It is created by cutting and splicing together DNA from different sources such as different organisms or different parts of the same organism. What is gene editing? Gene editing ante it is a technique used to make specific changes to DNA sequences. This can involve adding, deleting or modifying specific nucleotides in the DNA. What is cloning ante? Cloning is the process of creating genetically identical copies of an organism or a cell. This can be done through a variety of techniques, including somatic cell nuclear transfer, which involves transferring the nucleus of somatic cell into an egg cell that has had its nucleus removed. These concepts are the foundational to the genetic engineering and provides the basis for many techniques and applications that are used in the genetic engineering field. These basic concepts of genetic engineering. Thank you for watching this video. If you really like this video, please subscribe to Groups Master YouTube channel. Thank you.